All right. Hello. Welcome, everybody. Thank you for joining us. I'm Jasmine Navarez, Digital Content Coordinator at Simulations Plus. Nate Moster, Director of Alliance Management, will introduce our speakers for today. In this webinar, James Mann, Principal Scientist of In Vitro Product Performance, AstraZeneca, and Xavier Pippin, Associate Vice President, Regulatory Affairs, Simulations Plus, will discuss using GastroPlus, PBBM, PKPD to define dissolution, safe space, and support of registration specifications. A few housekeeping items before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. I'll now kick things off to Nate.
Thanks, Jasmine. And hello, everyone. Thank you for joining us today. I have the privilege of introducing our presenters, starting with James Mann, who is a principal scientist of in vitro product performance at AstraZeneca, with more than 16 years of experience as an analytical scientist specializing in all aspects of dissolution testing. At AstraZeneca, James is the global lead for the in vitro product performance scientific community, with oversight of all dissolution-related activities in product development. He also co-leads the International Consortium for Innovation and Quality in the Pharmaceutical Development Dissolution Working Group. Prior to joining AstraZeneca, James obtained his PhD and led multiple teams at Merck. Our second presenter is Xavier Pippin, who is a pharmacist with a PhD in granulation technology related to powder surface energy and liquid bridges during wet high shear granulation. He has more than 25 years of experience in the pharmaceutical industry and has occupied several positions from pre-formulation, clinical and commercial formulation development, industrial transfer, regulatory CMC, and biopharmaceutics. His work in biopharmaceutics over the last 15 years has been using in vitro, in silico, and in vivo tools to support evaluation of drugs along the development chain and post-marketing, with numerous publications across those areas. So, gentlemen, thank you again for presenting today. The floor is yours.
Thanks, Nate, for the kind introduction. So I'm going to share my screen. Let me know when you can see it.
Yep, we see them.
All right. So as we mentioned earlier, we're going to talk today about GastroPlus, about a physiologically based biopharmaceutics model, PBBM, and a PKPD, so a pharmacokinetic pharmacodynamic model, which were used in conjunction to define the dissolution safe space for a drug product registration. So I'll be speaking first, and then James will also present the case study.
Xavier, just so you know, you're in the presenter view, not the full slideshow.
All right. Settings. There we go.
That's it. Thank you.
That's it. Thank you. So in terms of outline, we will present some definitions. We'll talk about the scientific and regulatory landscape for PBBM. I'll present some industrial applications of this tool. And then we will delve into the case study combining PBBM and PBPK to develop and set the specification for a recent tablet formulation that was developed by AstraZeneca using GastroPlus for the spec setting, acalabrutinib maleate tablet. So let's start with some definitions. You're familiar with the term PBPK, maybe. If not, it stands for physiologically based pharmacokinetic model. It is an in silico tool that represents the physiology of human and animals in a mechanistic way, where all the tissues have real volumes and are equipped with fluid flow and enzyme expression. So you can use those models to do many things. You can predict DDI, for example, but you can also predict the limitations of drug absorption, and if you're focusing your work on that, you would call this model PBAM, physiologically based absorption model. And finally, if you use the tool for a biopharmaceutics model, so linking the quality attributes of drug products to the in vivo exposure, you would call this a PBBM. So basically the PBPK is the tool, and the PBAM and PBBM is what you do with the tool. I'd like to introduce as well the concept of safe space. So the safe space is this area here in green that you see on the screen. It's the space where all the batches that are defined by their variation of critical material attributes or critical process parameters are anticipated to be bioequivalent to each other. This safe space is really important to define as soon as possible in the development of a drug product, because you have some flexibility to change specifications, to navigate in your beyond the operating range that you may have defined for your product and your process, and still be bioequivalent to your clinical reference. At the edge of the safe space, we call this the edge of failure. So if you manufacture batches beyond the edge of failure, you would predict non-bioequivalence to the clinical reference. And the outer edge of the batch manufacture that you have tested in the clinic would be called the knowledge space. The last definition I will give you is about dissolution methods. There are important characteristics to dissolution methods. First of all, you would want to have the right discrimination level, meaning that if you observe a change in vitro, this change would translate in vivo to the same level of variation. So that would be the perfect case study. Some methods are deemed over-discriminative, so a big change in vitro would translate to a smaller change in vivo. These methods can be used to establish specifications, but they're not favored. And if they're too over-discriminative, if they are too permissive in terms of variation for the in vitro property, they may be rejected by regulators. So it's something that you can use in some cases, but that you have to use with caution. And finally, a dissolution method can be under-discriminative, meaning that a small change in vitro would have a big change, would translate to a big change in vivo. These methods are, you have to run away from these methods. They cannot be used to do anything meaningful, and they cannot be used to set clinically relevant drug product specification. I mentioned that methods can be discriminative, but there are two other qualities of the method. They can be clinically relevant, so they can rank order with the clinical performance. So batches that would dissolve slower would have a lower Cmax, for example, than batches that would dissolve faster. And they can also be biopredictive. So if they are biopredictive, you can use the dissolution data to integrate it into PBBM or an IVIVC and predict with sufficient accuracy the human PK. And that's really the methods we're after when we develop a PBBM, because we can then use dissolution as a surrogate for in vivo evaluation. You can use dissolution, and you can use the dissolution data in your model to predict human exposure, and therefore you can waive human testing. In terms of the biopredictivity or the clinical relevance, that can be applied to a QC method or to a biorelevant method. It doesn't matter. So the QC method would use a very simple medium, stable, easy to control USP apparatuses, for example, whereas the biorelevant method may include more complex media, may include different transition of fluid, an absorptive system, for example. So these are more complex to handle, but could be useful as a biopredictive method as well. Right, let's move to the second point of my presentation, which is the scientific and regulatory landscape around PBBMs. Look at the number of publications where the term PBBM has been used, and I apologize, I've stopped that search in 2021, but you can see there's an exponential growth of that term. And as well, looking at PBBM in conjunction with dissolution specifications, or PBPK in conjunction with dissolution specifications. So we are using these tools, PBPK and PBBM, more and more to probe the product quality and to understand the link between material attributes and in vivo performance, or process parameters and in vivo performance. And these tools are now a regulatory tool as well. So in terms of regulations, there are 3 main regulations that have been proposed by Europe and the US. In the US, there are 2 regulations. One which was back in 2018 regarding the format and content of PBPK analysis, followed by the European submission as well on the reporting of PBPK modeling and simulation. And more recently, the US has submitted in 2020 the use of PBPK analysis to support biopharmaceutics evaluation of drug product development. So this guideline, which has a very long title, I would abbreviate it by the PBBM guideline, even though it's not going to please our colleagues from FDA, but that's how we are basically. It's the first guideline that mentions this term and also mentions how we can use PBBM to support product dissolution, for example, or the setting of a drug substance particle size. And back at the beginning of 2021, Japan has also published a guideline to report PBPK models. And as you can see here, there's been quite a few workshops that have been organized and publications coming from those workshops as well, which detail the pros and cons of and the merits of different models, and also evaluate the requirements for model validation, for model use. So that's a very important aspect, because at the moment the guidelines are not harmonized. There's the criteria for accepting a model are not very clearly outlined. So there is more work needed, and this year in 2023, around August, there will be another workshop that will be co-organized by the IQ committee, and also will have participations from the regulators from the US, from Europe, from the UK, from Health Canada, Brazil, and Japan. So it will be an important workshop to attend to. So the guidelines, I've already mentioned those. You have the links here if you're interested. I'm not going to spend too much time on it. So these are the 3 US, Europe, and Japanese guideline on the format and content of submissions. And this guideline here is the PBBM guideline from the US, which defines the utilization of PBPK towards biopharmaceutics application. Now that we've seen the regulatory landscape and the scientific landscape, we can talk about some applications of PBBM. So first, I'd like to talk about the benefits of PBBM. They really bring a mechanistic understanding. I was mentioning earlier that the PBPK tool is made of real tissue volumes and real tissue functionality. So it is a tool that can be translated to a different population, and a very flexible tool, and very nice and mechanistic tool that was invented. And PBBM is exactly the same. You integrate drug dissolution in a mechanistic way, and you can really probe the limitations of drug absorption, for example, through oral route, or drug reaching the systemic circulation if you dose it in any non-oral route. Knowing this information, you can improve your formulation. So you can also guide formulators to have the first time right. So have a commercial formulation which is the best available for this drug. And you can also set, for example, acceptable thresholds for certain types of excipients using these tools. So it's a really important tool that can be used early on. And towards the end of the development, you can define the safe space and understand where you have to put your product specifications, and that will be very useful for the submission. And the example we have today will address that justification of product specification. Finally, post-approval, because you have manufactured as an innovator company or even a generic company, you've manufactured a limited number of batches before registration. You don't really know how your product or your process performs on the average, and you may have to be shifting some of the specifications you set at submission because they were too narrow. And the understanding of that safe space will allow some flexibility to the manufacturers, because they can shift those specifications within the safe space, and that would be acceptable without any human evaluation. And finally, there is an ethical aspect to conducting PBBM, because you can reduce unnecessary human testing. Some drugs are variable and they require a lot of subjects to be recruited to conduct a bioequivalence evaluation, and that can be unethical to submit a healthy volunteer to, for example, a cancer drug from which they will not benefit. So we can reduce with PBBM the need for clinical trials. We can also optimize the resources. It doesn't mean that no clinical trial have to be done, but you can conduct them in a wise way. By combining QBD approaches to your clinical evaluation, testing variants of your formulation that can define a large knowledge space. In terms of savings, they are quite important. I won't go to all of these details, but you can improve time to market from 1 to 2 years. You can also improve your yield if you have a product specification that was too narrow and you're wasting product based on out of specification products that you have to throw away. You can answer regulatory authorities in a very quick time. And finally, you can avoid doing bioequivalence evaluation. So overall, a consequent gain in time and money during your development. In terms of benchmark on how people in the pharmaceutical industry use PBBM, basically they are 1/4 of the application which is happening during development and 3/4 which is happening as a post-approval change. So it's a really important tool throughout the development timeline.
So Javier, can I cut you off? On the bottom toolbar, is it okay if you hit hide? It's kind of blocking your slides out. Yes, thank you.
Sorry. Is that okay? Is that better now? Sorry about that.
Yes.
Yeah, so as I said, this tool is really important across the development timeline. Very early on, you can predict human formulation that will be selected, whether they will be reaching the target exposure, whether you expect some variability and where does that variability come from, and that gives you the opportunity to improve it. And this is the same when you do due diligence. For the example that will be presented today by my colleague James Mann, we did a due diligence when AstraZeneca licensed the drug, and that due diligence was done with GastroPlus and revealed some drawbacks of the drug which were later improved. As the clinical phases progress, you learn from your phase 1, and that's the nice thing about PBBM and especially GastroPlus. You can integrate lots of different inputs from the learnings of your phase 1. As soon as you generate human PK, you can analyze it with a top-down approach, extract some information, or refine your predictions that you did prior to human testing, and that will reinforce the accuracy of your model. And of course, when you design your commercial formulation, you can go first time right. You can really improve the formulation compared to what you had in phase 1. Later on, you can inform your control strategy. We talked about this, and this will be the example presented today in the justification of a drug product dissolution specification. And finally, post-approval, you can also develop lifecycle management products, develop generic formulation, and also change your specifications without doing any human evaluation. Just a brief word before I leave the floor to James on why it is important to combine PBPK and PBBM. And sorry, PKPD and PBBM. There is a typo here, PKPD and PBBM. So PBBM, as you have seen from my presentation, links the drug product quality attributes to in vivo exposure. And in terms of quality attributes, it could be anything from product dissolution, drug substance particle size, polymorphic impurities, manufacturing process parameters, in vivo degradation, effect of excipients, etc. There's lots of things we can do, and ultimately the PBBM is validated against clinical data. You can use it to define safe space where all your batches are bioequivalent, and typically this is achieved through the running of what we call virtual bioequivalence studies, where you would maybe run 10 studies on n equal 25 or more patients or subjects and healthy volunteers, and then you would compare the geometric mean, calculate confidence interval, and use the standard B criteria or reference scale B criteria, depending on the variability of your drug, to conclude on the bioequivalence. So when you do this exercise, the size of the safe space is bound by your bioequivalence criteria. You have to be bioequivalent. If you look now at PKPD models, they link the product exposure to drug efficacy and safety. They can be based on observed data, just being simple mathematical relationship, or more mechanistic model based on the understanding of the mechanism of action, and could go all the way to cell signaling pathways. These models will therefore predict the effective space, not necessarily the bioequivalent space. And typically this is usually larger than the spaces you can get through just applying the bioequivalence criteria. So basically the combination of PBBM and PKPD, sorry, make a mistake again, would link the product quality attribute to the product efficacy. And this increases the spaces you can determine, the safe space you can determine for a product. Right, so that's the end of my presentation. I would suggest we keep maybe the questions for the end, and I will stop sharing and now ask James to present case study and a collaboration in Mali. Thank you.
Okay, I will just share. Let me get it to full screen. Can you just let me know that you're seeing it in full screen mode?
Not yet.
Okay. Hold on. Sorry, let's try sharing again. Okay. Is it coming through now?
Yep, it's coming through.
Excellent. So thank you very much for the opportunity to present our case study today, which, as Xavi has alluded to, is how we justified our clinically relevant QC specifications for one of our drug products. So just to quickly come back to something that Xavi said, is we obviously have a multitude of dissolution methods that we use in development, and there are two papers that are two of my favorite papers here that are cited, and they're both come out of the IQ Dissolution Working Group a few years ago. And the first one is really related to how we define dissolution methods. And obviously we have QC methods, we have physiologically relevant or biorelevant, sometimes called. We have the bio-waiver, SUPAC type multi-pH medias that we'll run. But we have this idea of clinically relevant dissolution methods and specs. And essentially, as Xavi pointed out, a QC method can be clinically relevant, physiologically relevant method can be clinically relevant, and that's really what we're after. And what that allows us, if we have that clinically relevant or biopredictive method, we can really then use that to drive our spec setting and our control strategies. And I guess this is the decision tree from the Herman's paper, which then focuses on setting the specifications. And in the old days, we tended to go down that left-hand side of the approach one, where we basically setted our specs on what we had dosed in the pivotal studies. nowadays, certainly at AstraZeneca, our strategy is much more focused on developing clinically relevant methods. And if we have variants in the clinic, and it says formulation and or process variants, it doesn't necessarily need to be that. It could just be you've dosed your product under different physiological conditions, be it PPI or food, and you can build a really good PBBM. You can probably push yourself down this approach. And there are multitudes of them. You can have the classical level A, level C, IVIBCs. These are obviously much harder to achieve for IR products than they are for an MR product. You can have the safe space that Xavi has outlined, where your dissolution method is probably over discriminatory to the in vivo situation, and so you have that space built in. And then you have this more in silico approach, in vitro in vivo extrapolation, or PBBM based model, as it's probably more commonly known now. And really our case study will focus on that side of the decision tree. And so, yeah, with the case study, it's based around the Calquence product, which is acalabrutinib, which is a selective irreversible bruton tyrosine kinase inhibitor, which we brought in from a company called Acerta Pharma back in February 2016 with the AstraZeneca team at the time, which was comprised of myself and Xavi and others, were responsible for all the CMC related activities of Calquence. And it was approved as a free base capsule in the US in 2017, and then in much other markets for a variety of blood cancers. And we developed a PBBM model for it at the time, and there are 2 papers that are pretty been out there for a while now, since 2019, where we have the first paper, part 1, where we really look into the dissolution and building a mechanistic understanding, and use something called the product particle size distribution, which I'll cover in a little more detail on the next slide, as the input into the PBBK. So it's that mechanistic integration of dissolution into the GastroPlus model. And then the second paper, which is more focused on the actual modeling side of things. So I'm not a modeler, I'm an in vitro scientist, as was made clear in the introduction. So if you have real in-depth questions on the models, then these papers will be really good references to the underlying model. So just to talk a few points about how we handled in vitro data and solubility data into the original Calquence model, is we did this use of this, what we term product particle size distribution, which is essentially a translational parameter in order to allow you to integrate the dissolution in a mechanistic way, where the model can then dissolve the particles that you're giving it based on the pHs and volumes, etc., within the model, the ACAP model. So you would tend to fit. In our case, it's a 10-bin particle size distribution with free optimization to a dissolution data. We will then validate that, and I'll come on to that in the next slide, as you have to make sure that that's a valid input into your model. So that's one element about how you integrate the dissolution. The second important input that we uncovered during the acalabrutinib story was really the importance of surface pH. So acalabrutinib is a dibasic product with 2 pKs in the physiological range, very steep solubility curve. And as you'll see here, the bulk pH, say in 0.01 normal HCl, pH 2, the actual surface pH of the acalabrutinib would actually be pH 4, and so that makes a big difference to the solubility value that's actually driving dissolution, and we will see that on the next slide. So here is the idea of validating the PPSD. So in this case, we fitted at pH 6.8, and then we basically did some experiments under other dissolution conditions of different pHs, some incorporation of some micelles. And essentially what we then did is we used that PPSD, and we virtually dissolved it under the various conditions, and that's the blue lines here, and you can see that looked pretty good relative to the experimental data, which is in the red. Now the blue line is if you use the surface solubility. The dashed line, which is the one that you can see clearly overestimates the dissolution under every situation, is if you use the bulk solubility. So it really is incredibly important for this molecule that you use the surface pH value. And then really just to touch, because this isn't really the focus of the talk, but the model validation of the original model. There is actually 16 different clinical scenarios that it was validated against, but I've focused in on the really important ones here, the 7 really important ones. So we had 2 really different independent studies, which we had an early and a late formulation. We had 2 different dosing scenarios for each of those situations, and they raised, one was under the normal stomach pH and one was under PPI treatment. And then we also had a situation within one of those studies where, in an attempt to overcome the PPI effect that we were seeing, they also tried to dose with an acidic beverage, and they also looked at grapefruit juice for the CYP3A4 inhibition. And so we were able to, as you see for AUC on the left and Cmax on the right, we really were able to predict the outcome of the clinical studies pretty well, and we were able to reproduce meaningful in vivo dissolution, capturing both the impact of the changes in physiology and the impact of formulation. But the story did not end there. The product was approved, it's on sale globally, but we knew there was a weakness in it, and this is, we had that the capsules had a restriction on relation to use with gastric acid reducing agents, mainly PPIs, and as you can see the actual label there on the screen, which says avoid co-administration with proton pump inhibitors and staggered dosing with H2 receptor antagonists and antacids. And this was seen as a downside. We wanted to increase our ability to reach more patients. We wanted to take that decision making from physicians away and just really know that they could provide, calquence could provide some exposure for everybody. And so a little bit serendipitously, the chemistry colleagues were actually working on an improved route design, which included going via a salt. So we intercepted these salts, and we were able to use the solubility data and the dissolution of these salts to input into the already validated PBBK, which we were able to then use that to really help select the correct salt. We also did a wider, biopharm isn't everything. We looked at the salt from process chemistry, formulation, solid state stability, etc. as well. And this is where the malate salt was identified. And that solubility diagram there just shows you the very steep solubility curve that you experience for calbutidine and why we were really having that observed PPI effect. So we used dissolution in the formulation selection. We were obviously, our key development aim was to overcome that PPI restriction. So we used medias that would represent that PPI treated stomach. So initially we used just water, but later on we used a PhD student of ours at AstraZeneca, Dom Segregur, who had developed some PPI H2RA media. And on this plot here, you can see that we were able to do dissolutions of drug substance, which would be the blue on the left, and then we were looking at disintegrant selection, and we saw that we were actually seeing a little bit of disproportionation in the presence of some particular disintegrants, cross-carbonyl sodium and SSG. That last time point is where we added some acid, so if there had been conversion to free base, we would recover it. So we were able to select the disintegrant here, and then in a similar way, we were able to look at the fillers, and we were seeing a little bit of disproportionation with the presence of dicalcium phosphate there, again, ionic excipients. So really, the dissolution was helping select the formulation. And then we moved on to the method development for the actual QC test. And we first of all started by screening the pH. And as you can see, 0.1 normal, the blue is the capsule free base product, the green is our prototype tablet at the time. And 0.1 normal, you can see they both look very much the same, which is what you'd expect. Probably converted all the salt back to free base at that. But as you moved up the pH into the 4.5 acetate, the water, and into the traditional USP 6.8 buffer, you can see that we were very much seeing an improved dissolution performance of the tablet and the malate salt. So we focused on that. We focused on the 6.8 dissolution, and we moved into some more medias. And at this point, we had 2 different particle sizes. The blue batches, that you can see with a smaller particle size, the green with a larger particle size. I think the two different batches are two different drug loads. But you could see we really didn't have a huge deal of discrimination for particle size under the 50 millimole phosphate. So we looked a bit wider. We reduced the ionic strength there to 5 millimole, and we started to pull out better discrimination, as you can see on the top left plot. We also saw that level of discrimination for the 50 millimole tris pH 7, but that's right on the buffer limit for tris. So we didn't feel comfortable with, from a robustness perspective, for that. So we focused in on that 5 millimole phosphate. Once you have that, then you obviously have to select the paddle speed. So we did the standard 50 V75 RPM, and at 50 RPM we did see some coning. So we focused in on 75 RPM as our QC method condition. What you then need to do is obviously assess the discriminatory nature of that. So we had some direct compression prototypes with 3 particle sizes, which you can see, and 2 drug loads, which you can see on the screen. That was showing a very nice discrimination. We also assessed other potential failure modes around accelerated storage, reduced disintegrant levels, increased lubrication. And obviously we did a DOE around the actual formulation and manufacturing process, where we looked at the ability to tolerate different APIs, alternative excipient grades. We started to move from a DC formulation to an RC formulation, and looked at those properties, blends, compression forces, etc. All the good things the formulators like to study. And so we've selected the 6.8 method as it's the most clinically relevant to ensure that our product will show good release at both low pH and the PPI treated. We're getting full release within a clinically relevant time frame. We were seeing very low method variability. We could discriminate for slow disintegrating tablets or large API tablets. We had good discrimination for other failure modes, and we had what's key here is you can't really develop an over discriminatory method and then have it basically unable to be run in a QC method. So it also had acceptable method robustness. So the clinical plan for this product to get it on the market was actually very straightforward. It was only 2 studies. We had a sort of pilot REL BA study that was split into 2 parts, and we had a formal BE study. The publication on the top right, you can see in detail the clinical design and clinical data for this. It's published, but essentially in the first part of the REL BA study, we had a crossover design in the fasted state between the tablet and the capsule in periods 1 and 2, and then as you move to period 3, we went to a fed study for the tablet and then a PPI arm for the tablet. And then part 2, we looked at a solution versus 3 particle size variants, which you can see in the top right plot there, the dissolution plot. So the green being the capsule that was dosed in this study, and then the 3 batches of tablets that were dosed. We were trying, had quite a wide range of particle sizes there, but once we moved to an RC process, we didn't see quite as much discrimination as we did for the DC, and also our chemists were not actually able to make us as large a large API as we were after. But needless to say, this is what we dosed. We were able to assess the impact of that on PK. And once we'd done that, we then did a formal BE study, just a crossover study in the fasted state. And you can see the 2 batches that were dosed there for the tablet and the capsule. And essentially the outcome of this was that we showed BE between the tablet and capsule in the fasted state. The tablet showed no significant PPI effect, which was really the quality target product profile for this development. There was a lack of food effect on the tablet, and actually there was a lack of impact of API particle size on exposure within the range that we studied. And this, I guess, is the key slide, is how did how did those clinical studies actually look versus the PBBM? Because we were very confident going into those studies that we would succeed. And as you can see, the Cmax is on the top, the blue is the measured versus the orange is the predicted for all those arms of those studies. And on the bottom is the AUC, and there's a little bit of statistics here. So the AFE was 1.05, with the average absolute prediction error was 8.5%, and the Cmax, the AFE was a little bit lower, 1.01, and the absolute prediction error was 6.7%. So a very powerful model, I would say. And how we then drove that into spec setting was because we hadn't got the particle size range that we had asked the chemists for, we didn't really, we thought we could have a bigger safe space. So we created 2 virtual batches, which are on this virtual batch A, which is the purple one, virtual batch B, which is the red one. And then essentially they run through the Q80 at 20 and 30-minute spec time points. We were then able to use, by converting them in through the PPSD approach, converting them into particle sizes and integrating them into the model, we were able to calculate the relative exposure to our reference in both acidic and neutral conditions. And in our original submission at the top, you can see that realistically in a fasted stomach, all virtual batch, all the way down to virtual batch B, the red profile was within the bioequivalence bounds, which the reference was TAB here, batch. And then in the pH 6 PPI treated stomach, you could see that for AUC, which is the left-hand side, virtual batch B was right on the BE bound for the mean, probably would be outside the BE bound for the confidence intervals. And for Cmax, both of them were outside of the BE bounds. But we weren't too concerned because we had BTK occupancy, so which is the PD element of this. We actually had physical enzyme occupancy data for this, which we could apply to the model, and that showed that we were actually hitting our target, way above 95% target engagement. So we were quite happy with this. We submitted it. The FDA wanted to squeeze us, I think, but they actually redid our modeling and they pointed something out to us, which was where we were using an individual, we were using TAB reference in the stomach, low pH stomach, but we were also using the reference for TAB in pH 6. What they asked us to do was actually use a single reference, which would be the TAB under the low pH as a reference, and that then when you redo those GMRs plots, which is what you see on the right, actually squeezes you into the 20-minute time point, which doesn't really make any difference for the fasted stomach. As you can see, you're still well within the BE bounds, but for the pH 6 stomach, it takes the virtual batch B away from that borderline AUC bound and puts virtual batch A right on that borderline. And so we were able to, accept 20-minute spec. That was still obviously way outside the bounds of what we had dosed in the clinic, but we were able to show through the BTK occupancy model that we were still in a massively safe space from that. And so, we got a 20-minute spec as opposed probably to being forced into a 15-minute spec, Q80 at 15 minutes, which is typically the tightest spec that you get pushed to. So with that, the FDA approved, after redoing all our modeling and having a bit of back and forward with us, they approved the Calquence tablets in the US in August last year. Subsequently, we've received further approvals in EU, Australia, Canada, and Brazil, and we will continue to seek more global approvals as we continue to roll this product out to replace the capsule product. All this work was actually published in February, and I'm sure as part of the webinar package, the link to the publication can be provided. This was just, I had these slides pre-approved, and so I was not able to add the link directly to the new paper. A few acknowledgements. Obviously, I'd like to heavily acknowledge Xavi, who in his past life was with me at AZ, has now moved on to the GastroPlus, Simulations Plus side of things. But yeah, like to highlight Natalie, Andrea, Rick, Alex, Anna, Vivian, and Louise from the AZ side, who've been heavily involved in the entire Calquence development. And then some colleagues from Acerta when we bought the molecule, in Cecile, Marilee, Greg, and Tim, who were instrumental in that early transition into the AstraZeneca portfolio. And with that, I'd like to thank you guys for your attention, and we can hand over for questions, I think.
Thank you, James. Thank you, Xavier. Before we do pass it off to Nate for the Q&A, I do have one more poll, and it is, are you planning to or have already incorporated a PBPK, PBBM simulation result to support regulatory submissions this year? And I'll give it a couple more seconds. All right, thank you. Go ahead and take it away, Nate.
All right. Thank you, and thank you guys for the presentation. There are a few questions that came through, so I'll start with the first one that was, it says, do you have any regulatory experience where bioequivalence failed, but PKPD indicated absence of clinical impact and the agency accepted?
So I guess we didn't technically have any BE failure, but we got a dissolution spec approved that would probably have been outside the BE bounds based on the PKPD argument. So I don't think we've done it when we've actually physically failed a BE study.
And I think this is a regulatory approach that is completely valid. You know, if you fail a BE study, it doesn't mean that you will fail the efficacy test. So you can definitely either run a study or use a PKPD model to define the efficacy of your drug, and that's what we did virtually with acalabrutinib maleate. But you can do it on any compound.
Okay, thank you. The next one says, how do you incorporate surface pH into GastroPlus?
I'll give this one to you, Xavi.
That one. So they are, first of all, I'd like to mention that DDD Plus does an estimation of the microenvironmental pH. At the moment in GastroPlus, it's not, this calculation is not made, so you have to do it manually. You've got two options. If you have, for example, a weak base where only the stomach is going to be impacted by the surface pH, you can just change the pH in the stomach and keep the bulk pH solubility profile as an input in GastroPlus, which is the standard pH solubility profile that you use by fitting the observed data of solubility versus final pH. Okay. That's the first option, and this is what we've done for acalabrutinib, the free base, because of its pKa. The strongest base is 5.8, and therefore at pH 6 in the duodenum, the base is already, is not ionized, and there's no acid-base reaction, so there's no impact of microenvironmental pH in the small intestine and the large intestine. So it's only happening in the stomach. So easy ways to change the stomach pH and then save your ACAT model as a specific physiology to your drug. If you have a very strong base, for example, pK of 9, you will have acid-base reaction happening in the duodenum and subsequent compartments. And in this case, the best scenario is to calculate the surface solubility versus pH profile, integrated as an SPD file in GastroPlus. You click on File, Load, SPD file, and then you can integrate your pH solubility profile. And when you run the simulation, you will have to use the SPD file to calculate dissolution. And then you can leave the pH in the ACAT compartments to their baseline value, to the physiological value. So these are the two options you have.
Thanks, Javier. A lot of good questions coming in, so I'll keep going. Obviously, if we don't get through them, they'll be available. But there's a question about, did you use the same PPSD in predicting dissolution in different media conditions? And how is bulk solubility calculated?
So I guess I've bring the slide up here. So yeah, we used the fitted, we fitted one pH and then we predicted other pHs to ensure that's valid. So we use a single particle size distribution to describe a batch. So if you have a different batch with a slightly different dissolution, you may want to refit. So it's very specific to the to what you were dosed in those specific clinical studies. The bulk solubility, we measure that. So we just do typical shake flask type experiments.
And just to mention, we did the same approach for acalabrutinib maleate. We had verification as well. So it's really important to do the verification, and it's important for the PPSD, but it's also important if you use a different mechanistic model, like the Z factor, for example. you can do exactly the same. You can verify that your Z factor can predict a different pH. So it's also recommended to generate dissolution data at multiple pH, multiple conditions, so you can actually have that verification available.
Okay. How do you assess the bioequivalent sample size and include WSV?
So the sample size is really important as for any clinical trial. So you would look at the within subject variability, try to do some calculations or ask your favorite clinician to do them for you. And basically you would scale your virtual B the same way as you scale a bioequivalent study in human. So here for acalabrutinib, for example, we had 66 subjects for demonstrating bioequivalent, because this drug is borderline highly variable. So we had to resort to a large number of subjects. And if you do a virtual B, you would do exactly the same. You would select the same number of subjects to be able to run the VB. The other thing that you can check in GastroPlus is, and I would recommend you do it systematically, is once you have your reference batch, for example, a phase 3 clinical batch as an input in the model, the first virtual B you do would be this batch against itself. So you would verify in a crossover study that you can demonstrate bioequivalence with the same batch given twice to the same subject in a crossover B. Sometimes you may have, and in doing so, I would suggest you tick the option, use physiologic intra-subject variability, which will slightly change the transit time, the pH, the volumes, and the bile salt concentration in your ACAT model, because this will give you a realistic view of the potential within subject variability and how that impacts the drug. So depending on the type of drug, it may be a big impact or it may be a small impact. For example, for acalabrutinib, the biggest impact for the freebase was the pH in the stomach, not necessarily the bile salt concentration in the small intestine. But for other types of drugs which are more lipophilic or and more sensitive to the presence of bile salt, maybe that parameter would be the key parameter to define your within subject variability. So that check is a sanity check. If you have adapted your model to your clinical variability, if you have population studies that are a good representation of both the average exposure but also the 90% confidence variation, you may be in a good position to run your VB, but that sanity check is always recommended. Test the reference batch against itself and see if you have to reduce or increase the variability to pass or that batch. Mostly going to be reducing the variability.
All right, I think we got time we'll do one more. You guys will be busy afterwards with some of the follow-ups, so spurring good Q&A, which is good. And this one maybe is a little broader, but would love to get both your thoughts on. It says, other than time, what is needed to gain greater appreciation and adoption for the value of PBBM, both in pharma and with regulators?
So I can start and maybe James, you can continue. So I think it's important if you work in the pharma industry, whether you are working for a small or a large pharma or for an innovator or a generic company, this is a new tool, okay? It's a new way of using an existing tool, which is PBBK, but PBBM is relatively new. The term was invented in 2019. And it will take time for people to realize the benefit of this tool and adopt it and make it their own. And when I was in AstraZeneca with James, we were lucky because we were followed by our management, we were followed by our regulatory CMC function, and they didn't blink twice when they were about to submit full PBBM to FDA for an accelerated review for calquence. You know, we went out there, we had discussions with FDA, we had TCs with FDA, we took on their comments, we updated the model for use for calibration malleate. So it's really important to go out and discuss with regulators, because regulators are scientists and they will come back with good comments, good suggestions, and that's something you can then integrate and improve your model as a consequence. So, you know, having faith in your scientists internally and having the courage or the ambition to go out and discuss those models is needed, and you know, it's a call really for every person in every company to you know, apply this to your own projects and go out there and present it and discuss it, publish it. It's really important to get that visibility, because there are a few numbers of examples, and that's really the spirit of the workshop we are co-organizing with IQ in August, really to have multiple examples from different companies to discuss the pros and the cons, the merits and the things that could be improved, because ultimately we're all learning, and the guidelines are not harmonized. So it's really the right time to do this exercise. James, you want to add something to this, maybe from your experience?
I think you've covered it really well, Xavi. But I think it's publication. You know, I think it's publication and presentation. You know, you have to show off the science. You have to let people scratch underneath the hood and really see what's going on. And so I think you can trace back the malleate, calibrant and malleate story all the way back to the Zorampic Lazinurad paper that Xavi published, you know, a number of years ago, which was the first time we really used the PPSD approach. And then we've obviously published the two calquence papers I cited at the start, and then as you search, you'll find the calquence malleate paper from February this year. So there's just this continued thread where we're being quite open and honest, and to be honest, to be fair, you have to get your management support for that, because, you know, people could see this as a competitive advantage. You know, they may not want the generics to get their hands on it. And I have had comments from some colleagues at other pharmas which says they really like these papers because they can sit in a workshop, your environment, and a training environment, and they can use the models and they can recreate what was done. And I think if you can take some information, you can recreate what someone else has done and get the same result, I think it gives you confidence that, you know, you're actually using the tools correctly, and then you can then apply that to your own project. So I do think it's just probably for the next sort of handful of years, we probably have to be very proactive as an industry in sharing our examples, and then maybe we can lock it down a little bit and start using them as more competitive tools once the regulators are fully on board with them. But I think, you know, we need as an industry to move forward together, and that's why things like the workshop that in August that Xavi refers to with IQ is really key, because I think there are, you know, 4 or 5 regulators who've been given case studies, you know, and they've gone away and they've reviewed them independently, and then they will come together with feedback on those case studies for the industry to take on board.
All right, great. Yeah, thank you both for your insights on that, and you know, doing your part to show some of the things that are going through these webinars as well. So really appreciate that.
Can I just say as well that you know, I think there's a question about the PBBM and other regulatory authorities. I think we've submitted it everywhere that we've so far submitted. We might take it out for some of the smaller nations, but all the large ICH nations are getting exactly the same file. That's not to say they've reviewed it in the same way that the FDA have. Maybe, you know, they've looked at it and just sort of gone, oh, your batch data says you can have 20 minutes. And anytime we've been pushed that way, we've also gone back with a response saying, yeah, we'll slide in from 30 minutes to 20 minutes, but here's the reason why, and it's the model that's driving this, not the, you know, not the we changed the reference as per the FDA request, and, you know, that pushes you into the 20 minutes. So we've never just said, okay, we're just going to come in and tighten just because you told us to tighten based on batch data. We've said we're tightening because the model says we should tighten, and, you know, that then educates them as well.
Yeah, fair point, and good with the information sharing. So appreciate that. Like I said, I think there'll be we'll do some more Q&A online afterwards, but I'm going to go ahead at this point and say thank you, gentlemen, and hand it over to Jasmine to close this out.
Thank you, Dream, Xavier, and Nate. Really great insight. Just for any of the questions we weren't able to get to, our BD team will reach out to you with answers shortly after the webinar, and then we invite you to learn more on our website. I put some links in the chat that you may find useful. Thank you again. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today.